Continuing on to talk about the cell theory and eukaryotic cells, we've talked about prokaryotic cells and we had a little quiz about prokaryotic cells. So now we need to talk about eukaryotes. And in general, we'll talk about eukaryotes and then we'll talk about specific eukaryotes after that. So we begin by talking about the major differences between a prokaryotic and a eukaryotic cell. Listed below each of the different types of cells are things that you should know, be able to recognize. So prokaryotes have no nucleus and they have no membrane enclosed organelles. They only have ribosomes inside their cytoplasm. There's nothing inside the cytoplasm that has a membrane around it. They have a single chromosome located in the nucleoid. And remember, the nucleoid and the chromosome is attached to the cell membrane, to the plasma membrane. There's no streaming in the cytoplasm. That means the cytoplasm doesn't move. It just sits there. They undergo cell division without mitosis. That is, they undergo what we call binary fission. They have very simple flagella, sometimes one, sometimes multiple, but they're very simple. They have smaller ribosomes. They have a very simple cytoskeleton, only to give it form, not to move the cell around. There's no cellulose in the cell wall. Remember, their cell wall is peptidoglycan. And they have no histone proteins that the DNA will wrap itself around. Compare that to the eukaryotic cell, which has a nucleus. They have membrane-bound or membrane-enclosed organelles. They have paired chromosomes. It means they've got 2 copies of each chromosome. They do have cytoplasmic streaming, so they can move things around inside the cell. They undergo cell division by mitosis, not binary fission. They have very complex flagella that have little molecular motors that run them. They have larger ribosomes. They have many structures that make up their cytoskeleton, the skeleton inside the cell. They have those eukaryotes with cell walls, have cellulose in their cell wall, with the exception of fungi, some fungi, which would have chitin in their cell walls. And they have DNA that's bound to histone proteins. You will have to recognize this for the quiz. So again, we've talked about bacteria and archaea. Those are good examples of the prokaryotes. In fact, that's the only 2 I can think of. We can throw in cyanobacteria, but cyanobacteria is just another form of bacteria. Eukaryotes are protists, fungi, plants, and animals. So it's important that you know that the eukaryotes consist of protists, fungi, plants, and animals. The plasma membrane of these eukaryotes encloses the cell, goes around the cells. In plants, they have cell walls made of cellulose again, very important. Plant cell walls are made of cellulose. And some fungi have cell walls made of chitin. So it's important that you know that cell walls can be either plants or chitin. Plants do not have chitin cell walls, but some fungi do have chitin-containing cell walls. Inside the cell, there's a fluid region that we call the cytosol. The cytosol is included with the organelles to make up the cytoplasm of the cell. So the liquid, the fluid, the gel-like part is called the cytosol, and the organelles that are inside the cytosol, along with the cytosol, make up the cytoplasm. So the cytoplasm is the gel-like part and the organelles. The gel-like part, the fluid part, if you will, kind of thick fluid, is called the cytosol. Now, eukaryotes do have a nucleus. The nucleus is a membrane-enclosed region that has the DNA and the associated histones. It also has a region inside of it that's called the nucleolus. So if you look here, this is the nucleus, this little dark staining area inside the nucleus. So the nucleus is enclosed inside something we call the nuclear envelope. And inside of that nucleus are the chromosomes, which are made up of DNA, and a protein that we call histones. Now, continuous with the nucleus is something we call the endoplasmic reticulum. So if you look at this illustration here, the nucleus here has it's got a double membrane system, nuclear envelope, and extending from that are the endoplasmic reticulum. Now, closest to the nucleus is something we call the rough endoplasmic reticulum. The rough endoplasmic reticulum has ribosomes attached to it. So you'll often hear me talk about the RER. The RER is the rough endoplasmic reticulum. And the RER very often always has ribosomes attached to it. And so at the rough ER is where these proteins begin to be made. Because remember, the ribosomes make proteins. The smooth ER is different than the rough ER because its primary function is to make and transport lipids, the cell membranes in effect. Okay, so that's important. The rough ER here is used to make proteins. The smooth ER is used to make lipids and to move them around in the cell. Now, very often attached to the rough endoplasmic reticulum, like I just said, are the ribosomes. Remember, the ribosomes are used to make proteins. That's their job. Okay, so the job of the ribosome, which is attached to the rough ER, is to make proteins. Now, just like in bacteria, there are some ribosomes that are not attached to the rough ER, and we call those free ribosomes. So the ribosomes on the rough endoplasmic reticulum are used to make proteins that are going to be included in a membrane, included inside a vesicle, and inside a membrane enclosed structure, or may be destined to go outside of the cell, or may be included in the plasma membrane. So you need to know those four things. I'm going to say them again just so that you have time to write them down. The endoplasmic reticulum, the rough endoplasmic reticulum, has ribosomes attached to it. The ribosomes will make proteins that will do one of four things. They will be either included inside of a membrane-bound organelle, included inside of an organelle that's inside the cytoplasm, such as the lysosome here. So it may make a protein that's included inside of this organelle, like in the lysosome. It may make a protein that's meant to be exported to leave the cell. So it can include something that's going to be in an organelle, or it can produce something that's going to be exported outside the cell. It may be used to make a protein that is included in the cell membrane. So it'll be processed through the smooth ER and become actually part of the cell membrane. Or it may also be used, included inside one of the organelle membranes. Now, so we've talked about the organelle. So kind of like a factory inside the cell, the nucleus will make the RNA that's going to be turned into a protein, and the protein is going to be produced inside the rough ER. Then it's going to be moved from the rough ER to the smooth ER. And in the smooth ER, it's going to be included, it's going to be processed and included inside a membrane that's being made or something. And then from there, it's going to be sent to what we call the Golgi apparatus. And the Golgi apparatus is kind of like a stack of pancakes. And inside the Golgi apparatus, any changes that need to be made to the protein are done there. So maybe I need to add a sugar to a protein, or maybe I need to change its shape. That'll all be done inside of the Golgi apparatus or the Golgi body. Okay, so kind of as a take home, the the Golgi apparatus is responsible for assembling and editing proteins. Now the proteins are moved from, it's inside the ER, smooth and rough ER. Typically those proteins are just sent through the lumen or the interior of the ER. But to go from the endoplasmic reticulum to the Golgi apparatus, there's little bitty kind of bubbles, little bitty membrane-bound structures that we call vesicles. And the vesicles will bring the protein, the processed protein, from the smooth endoplasmic reticulum to the face, the side of the Golgi apparatus that's closest to the nucleus. It's important that you know that face, that side of the Golgi apparatus that is closest to the nucleus is called the cis face, and you spell that C-I-S. The cis face of the Golgi apparatus is closest to the nucleus. The face of the Golgi apparatus that's closest to the plasma membrane is called the trans face. So cis is close to the nucleus, trans face is close to the plasma membrane. So after the protein's been altered and fixed and assembled and ready to go, there will be a vesicle that goes from the trans face of the Golgi apparatus to the membrane, to the plasma membrane. If the protein is going to be released, it's going to be pushed outside of the cell. If it's going to be included in the membrane, it's going to be that new membrane will become part of the plasma membrane. So I may ask you to trace the path of a protein, and you would say, well, it goes from the nucleus to the ribosomes on the rough endoplasmic reticulum, then through the smooth endoplasmic reticulum, then to the cis face of the Golgi apparatus, to the trans face of the Golgi apparatus, and then to the plasma membrane or to the cytoplasm. So again, let's take this a little bit slower so that you have time to write it down. The path of a protein production it starts in the nucleus. Then the messenger RNA is sent to the rough endoplasmic reticulum, where the ribosomes will begin to make the protein. So the nucleus to the ribosomes on the rough endoplasmic reticulum. That protein then is processed through the rough ER to the smooth ER, where it's either included inside of a vesicle or inside of a membrane. Then it goes using a vesicle, a transport vesicle. It will go from the smooth endoplasmic reticulum to the cis face of the Golgi apparatus. Nucleus, ribosomes on the rough ER, smooth ER in a vesicle to the cis face of the Golgi apparatus. Then through the Golgi apparatus to the trans face of the Golgi apparatus. Then from the Golgi apparatus either to the cytosol or to the plasma membrane. You need to know that path. You need to be able to either write that path down for me or recognize that path if I ask you a question about it. So again, one more time, just to make sure that we have it. Nucleus, ribosomes on the RER to the smooth ER in a vesicle to the cis face of the Golgi apparatus, through the Golgi apparatus to the trans face of the Golgi apparatus, included in the vesicle either to the plasma membrane or inside the cytosol, or the cytoplasm, to be more precise. Okay, now another organelle that you need to know about is the lysosome. The lysosome is like the cell's stomach. It's got enzymes that'll break down the used up organelles. It'll break down any other proteins that maybe were ingested by the cell. So it's kind of like the cell stomach. So the lysosome, a couple of important things that you need to recognize is that it does contain enzymes, okay, and those enzymes will break down and recycle cellular components or digested or ingested structures, like maybe a bacteria that was taken up by a macrophage. Now, very, very important organelle is our mitochondria. This is where we make ATP. Need to write this down. Make sure that you know it. ATP is the energy currency of the cell. Again, ATP is the energy currency of the cell. And ATP is made inside of the mitochondrion. This is the mitochondrion. It's got two membranes. It's got an outer membrane and it's got an inner membrane. And that's very important. We're going to talk a little bit later about how they make energy, and how they make energy is making use of both of those membranes. So important to make note that they've got both an outer and an inner membrane. So the mitochondria, which is where ATP is made, and remember that ATP is the cellular currency or the energy currency of the cell. And that mitochondria has two membranes, an outer membrane and an inner membrane that's kind of folded a whole bunch of times. Okay, so the mitochondria is where we make ATP. ATP is it provides our cells energy. It's the energy currency. And the mitochondrion has two membranes, an outer membrane and an inner membrane. And that inner membrane is highly folded. All these things are important that you know. Another organelle are the flagella and the cilia. They're very much like the flagella and similar to the pili in bacteria. They're used to move the cell or to move things around the cell. So the flagellum is a long, skinny whip-like structure that spins around and moves the cell through the aqueous environment. Cilia don't move the cell; they move the fluid around the cell. So, in your nasal passages, you have lots of cells that have cilia, and that cilia is used to bring mucus that your nose forms. It moves that cilia down through your esophagus inside your stomach. That mucus is secreted by your nasal cells to trap any foreign substances that get in there. So you would get sick a lot more if you didn't have that mucus there to trap those bacteria and other things that get into your nasal passages. And the way that works is that mucus traps that stuff, and then the cilia moves that. They move back and forth, and they move that. They move that mucus down through your esophagus to your stomach, so that those invading organisms can be broken down. So flagella again move the cell, and the cilia move the environment around the fluid or the aqueous environment around the cell. Now before I move on, there are some cilia, and especially things like protists, single-celled eukaryotes. There are some cilia that actually move the cell. There are some cilia that move, especially in things like paramecia and other things that actually move the cell. So I don't want to say that they're limited just to moving the aqueous environment. Now, just like your body has a skeleton, eukaryotic cells have a skeleton as well, and theirs is pretty complex. Their skeleton is made up of two different types of proteins. One of them called microtubules, the other one called microfilaments, and actually there's a third one that's called intermediate filaments. But for now, just know that there's microtubules and microfilaments, and they form the skeleton. We call it the cytoskeleton because cyto means cell. So this cytoskeleton is made up of microtubules and microfilaments. And they form just like your skeleton forms a supporting structure. They form a supporting structure for the cell. And actually they can be arranged or rearranged so that they can sometimes change the cell shape. Now in those cases, typically we'll see that mostly in things like phagocytes, which are cells that eat other cells, or we'll see that in especially protists. But they can be, I think in one of the videos I'm going to show you, there's a picture of a video of an amoeba moving, and the amoeba moves by breaking down the microfilaments in the cell and then rearranging them and pushing the cell membrane out. So the formation of this cytoskeleton inside of a eukaryotic cell is very dynamic. It changes. It changes all the time. All right, now, plants contain these big organelles that have enzymes and pigments in them, and they're called vacuoles. Now, animals don't have vacuoles. Only plants do, right? And in particular, plants have this big vacuole. It's called the central vacuole. And this guy is used in many cases to maintain the shape of the plant cell. Okay? So these vacuoles, whether it's the central vacuole or other vacuoles like peroxisomes or things like that, those things in plants are different than in animals. There are some that have enzymes and some that have pigments, some that are just storage. There are special structures that hold, for example, starches and things like that. Okay, but the one vacuole that I want you to know about, I want you to make sure that you know about the central vacuole, which serves to maintain plant's shape, and also to contain some enzymes and pigments. But be sure you know about the central vacuole. Again, when I stress something like this, that's really important. So make sure that you write these things down when I stress them. Chloroplasts, very similar to mitochondria. Now, plants also have mitochondria, but chloroplasts, remember I talked about mitochondria having two membranes, one membrane inside of another. Chloroplasts do too. When we talk about how plants make sugars, I'll talk about why it's important to have those two sets of membranes in them. Just like when I talk about how mitochondria makes ATP, I'll talk about how important it is to have two membranes. So these chloroplasts have two membranes, and inside the inner membrane, they have little pigments, lots of different pigments, but the important one, the one that's most important, is the chlorophyll. Okay, so plants have chlorophyll inside of their chloroplasts, right? And that chlorophyll is what the plants use, is how the plants get the energy they need to make sugar. So again, chloroplasts are part of the photosynthesis process, and they're embedded inside the inner membrane, the chlorophyll pigments that make this sugar are embedded inside the inner membrane of the chloroplast. There's your take home for that one. Now, real quickly, let's kind of go over this comparison between plants and animal cells. Plants have this large central vacuole that animal cells don't. Plants have chloroplasts. Animal cells don't. Plants have a rigid cell wall made of cellulose. Animal cells don't. Now that's plants and animals. Protists and fungi are something different. But right now, all I want to do is make sure you understand the difference between plant cells and animal cells. Again, big differences. Just the three kind of take home. Large central vacuole in plants, chloroplast in plants, rigid cell wall made of cellulose in plants. All three of those are lacking in animal cells. So you need to write that down, right? Large central vacuoles in plants, cell wall made of cellulose in plants, and chloroplasts in plants. None of those three are found inside of animals. Now, that concludes this lecture. I only want to go over plants and animal cells in a little more detail for the next lecture, and then that'll finish the discussion of eukaryotes. But remember, this quiz is going to include the cell theory, the differences between plants and animal cells, and the organelles of cells. So it's going to be a significant test. So you need to start studying now.